Today we will continue our discussion about the CAR-T cells from different sources and its anti-tumor response, and find out the CAR-T from which source performs better or have the better anti-tumor response. In the previous video we have understood the different sources of CAR-T and how they have uh, developed their CAR-Ts. So after that, what I did is, in order to check the CAR-Ts, the functioning of these CAR-Ts or the anti-tumor response of the CAR-Ts, first they did this. First they checked the different kind of CAR-Ts, that is different generation of CAR-Ts in in vitro conditions by stimulating them with their antigens to find out which CAR-T construct works well, and they are able to find out that the third generation CAR-T with this CD28 this CD28 BZZ, that is CD1928 BBZ, works well. So they decided to continue with this third generation of CAR-T to check the anti-tumor response of CAR-T from different sources. And they use three different conditions. First, the control, that is SK Hep factor, that does not have the antigen, and this another one is K562 cell line, and third one is the control, the SK Hep GPC3. And they also used other non uh, antigen cell lines, that is K0C, KOC7C, and HepG2. And they used the three different CAR-T from three different sources. First, from the immature CAR-T from the induced pluripotent stem cells. The second one is the mature CAR-T from induced pluripotent stem cells, and third one is the CAR-T generated from PBMCs. What they are able to see is that this CAR-T CAR-Ts that are formed from immature induced pluripotent stem cells towards T cell lineage shows cytokine productions in almost all the conditions. It is independent of whether the antigen specific cells are present or not. Whereas the other two CAR-Ts, that is CAR-T from the mature T cell lineage from induced pluripotent stem cells and the CAR-T from PBMCs, shows signals higher in the case where it is co-cultured with the cells that has the antigen, that is GPC3. So all these CAR-Ts that are used in their studies are designed against this GPC3 antigen. So it shows signals. So the immature CAR-Ts from the immature induced pluripotent stem cells show signals for all, whereas CAR-Ts from mature induced pluripotent stem cells and the PBMCs show signals when they are co-cultured with the cells that has those antigens against which these CAR-Ts are designed, that is GPC3. The quantifications are shown on the second row of the panel E, where we can see that the signals for the CAR-Ts from the immature induced pluripotent stem cells differentiation to T cell lineage is high in all the conditions, whereas the CAR-T signals from the mature T cell lineage and the PBMCs are high when they are co-cultured with the cells that has those antigens, that is GPC3, against which the CAR-Ts are designed. Thereafter, they also did the in vivo studies where on the one side they transplant a vector SK-HEP or the control tumors that does not have the antigen, and on the other side they transplant the tumors with the antigen specific tumor that has this GPC3. And then 14 days after their transplantations, they inject the different CAR-Ts, and what they're able to find out that the CAR-Ts from the immature T cell lineage from induced pluripotent stem cells does not give signals neither for the tumors with antigen present in it, or neither for the or nor for the tumor that does not have any antigen. Whereas the T cells or the CAR-T cells with from mature T cell lineage shows some signals for the subsignals at the site that has tumors with GPC3 antigen, and however the CAR-Ts from the PBMCs shows very high signals against the tumors at the site, tumors at the site where tumors with GPC3 antigens are present. Yeah, this is the correct way of presenting the results, that so CAR-Ts from PBMC shows very high signals at the tumors that contains these GPC3 antigens against which these CAR-Ts are designed, and the quantifications are shown in the panel G, where we can see that the signals from the CAR-Ts from PBMCs is significantly higher compared to the case where signals from the CAR-Ts that are derived from iPSC's differentiation, or induced pluripotent stem cells differentiations. So from these results, it seems like the CAR-Ts from PBMCs works well compared to the CAR-Ts from induced pluripotent stem cells. You will see in the subsequent videos what are the other things the researchers tested and what are their final conclusions. So far, the CAR-Ts from PBMCs seems to have better anti-tumor response than their other counterparts. So that's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.